Okay, I'd like to welcome everyone today to the first module of the CompuCell course. This is going to be a quick overview of CompuCell along with a few simple exercises that we're going to do together to familiarize ourselves with the usage of CompuCell. The links and the exercises are available for your viewing in the tiny URL CC3D 2025 links and exercises. And Hayden will also add the links as needed to the chat. As always, I need to remind you that this document and this session are live streamed and recorded and available publicly on YouTube. We may run over a little bit today since we're getting a slightly late start. I'd like to thank our funding sources from the NSF and prior funding from NIH. And I also would like to acknowledge the IUB Biocomplexity Institute, OpenVT, and Glimprint for their support. The Zoom logistics is going to be that we are going to have screen sharing, annotation, and microphones disabled during the sessions, unless you request assistance, and in breakout rooms. If you have any questions or needs, please put them in the chat, and please do remember, I think everybody's done that, to make sure that your name is your screen name in the Zoom. At the end of the sessions, we're going to do some polls, and there may be completion polls for the exercises as well. And all of the videos and PowerPoints are going to be made available to you as we go forward. If we're out of date, please let us know, and we'll do our best to update things as we go. The learning objectives for this early exercise, we've had Hayden already introduce people. I'm going to introduce them again briefly. I'll talk a little bit about the hackathon, and I'm going to then talk about how to use CompuCell3D after introducing virtual tissue modeling a little bit. And then we're going to run an angiogenesis demonstration simulation, learn a little bit how to control simulations in Player, the way you run things in CompuCell. And then we will use TwidIt, the CompuCell editor, to create a simple tumor simulation. We'll learn how to name simulations, define properties, and then we're going to run through very, very quickly CompuCell model specifications. And you'll see these things again and again. So don't be too concerned if you can't do everything that you need to do online. But we're going to do our best to give you a quick introduction to things so that as you come back to them later in the day and the week, they seem familiar. Logistics, as Hayden already said, Hayden has been doing the main organization. We're going to have lectures from people in a variety of institutions. Pedro del Castel and Gilberto Tomas are going to be doing much of the lecturing this week. We'll have lectures from TJ Siegel, Lorenzo Vischini, Jib Sluka, and Priyam Adyapok, among others. We're going to have several people who are going to come in and help from time to time as teaching assistants. Again, if you have questions, please email Hayden Fennell or ask in the chat. Typically, we're going to have half an hour before things start formally, when you can ask questions, have troubleshooting, and so on. We'll have an hour and a half module at the beginning. We'll try to make that partly lecture, partly hands-on exercises. We'll have about an hour break. And then we may have an extra module just before lunch where we discuss things like the hackathon. Then we'll have, again, an hour and a half module in the early afternoon, half an hour break, another hour and a half module, and then an opportunity for questions and comments. I encourage people to think about joining the hackathon. We can help You don't have to have had prior computational biology experience to be a participant in the hackathon. We can help you assemble a team. If you have a hackathon idea, we would like you to upload this 2 slides, 1 or 2 slides for that, and present that in one of these lunchtime sessions. Please contact Hayden about how to do that. If you don't have an idea, we can help you join a team. It's also a good idea as a learning experience to replicate existing simulations, and we can help you find one that's reasonable for you. And it's a lot of fun. We hope you'll join us. We need your input as we go through this course. CompuCell has a Reddit for discussion of bugs, problems, and feature requests. Go to www.reddit.com/r-CompuCell3D. If you find something that doesn't work, that's missing, documentation that's incorrect or confusing, you have an idea for improvements, please do let us know. If you have problems with things like crashes or issues, it's really important that you provide detailed information to us. Give us a zip code of the code you were running. Tell us what kind of computer you were on, what the operating system was, what the CompuCell install was, and ideally give us a screenshot of any error messages that show up in Player and in the Python console. It's very hard to debug things if you just say, CompuCell crashed. We need as much information as possible to locate the problems at hand. If you have a suggestion for the documentation, please give us the URL of and a screenshot of the page, and we'll do our best again to fix that. We have about 100 people registered for the class. We have about 60 people here today. We'll do our best to support each and every one of you, but we understand that that may not always be possible instantly, so please be patient. We'll do our best. If you're experienced with CC3D and you want to help beginners, there are people who sometimes come for the class and have a lot of experience. Please let us know. Again, contact Hayden Fennell if you want to do that. The workshop resources, there's the CompuCell website, the CompuCell downloads, and the CompuCell online manuals. It's very helpful to print out the quick start materials, have those in hard copy on your desk before you go forward. We've got the Slack channel, which is 2025 CC3D Slack this year, the Google Course Drive, Student Mats 25, and the CompuCell website, Workshop 25. If those are updated, Hayden will give the corrected links in the chat. Again, the Slack can be quite useful. Recommend people join it if you're not in it already, but do remember that Slacks are not permanent, and so anything that goes in the Slack will disappear at some point after the end of the workshop. And so if you want to make those things permanent, make sure you make copies of your discourse. The quick reference guides. There is a Python cheat sheet for people who want refreshers of Python. There is a guide to CompuCell3D. If you find things that are missing or out of date in that guide, please let us know and we can update that live for you. And there's also a sheet on tellurium antimony for network modeling. Again, if you find anything that's not right, please do let us know and we'll do our best to update those in real time. There are a couple of background readings. There's a PLOS paper by a group of students from Canada from a few years ago, talking in general about how to write mathematical models that could be interesting to people. There are 2 articles on the guts of CompuCell3D, which you can look at as well. Nobody comes to this class knowing everything about all the things that it does. Some people have more experience in wet lab biology. Some people have more experience in mathematics. Some people might have more experience in computing. You don't need enormous depth in any one of those areas, but you do need to know quite a bit about each one of them. And I'd say a Wikipedia level knowledge of each one is a good place to start. If you want to learn some Python, we like the Rosalind Python exercises. Network modeling, they're great Tellurium demos in the Tellurium Read the Docs. We have a couple of mathematics textbooks, which we like. There's the Essential Mathematical Biology. There's also the Mathematics of Life, which actually is a beautiful textbook. And I might recommend even over this Essential Mathematical Biology one now. If you want to learn some background biology, Gilbert's Developmental Biology is a great place to start. You can find an old copy relatively inexpensively. There's some good physics textbooks as well. There isn't really a textbook on this kind of multicellular modeling. We need to write one probably. We'd appreciate your suggestions for sources and reviews of these ideas. Anything that you suggest we should add will do. Let's talk for a few minutes about background and motivation for why we're doing this kind of multicellular agent-based modeling in the first place. Our world of questions fundamentally is almost all of biology. How does a fertilized egg develop into an adult organism? How does that organism maintain itself? That's homeostasis. How do failures of homeostasis lead to disease? That's called developmental diseases. The classic developmental disease would be cancer. We've studied infectious diseases with this kind of modeling methodology. People have used it to try to develop medical technologies for bioengineering and tissue engineering. It's also been used a lot in the context of toxicology. All of these problems depend on a complex interplay of physical and biochemical mechanisms. And that interplay between physics and biochemistry is really what you want to do with this kind of modeling. Computational models are really essential to translate the parts list that you get from omics and microscopy images into dynamic models that can give you understanding. This is a slide based on slides of Paul Macklin focusing on cancer. He calls this multicellular systems biology. If you look at tissues, tissues have an amazing array of structures. They're all built out of cells, extracellular matrix, and extracellular fluids. And we'd like to understand how these systems organize and sustain themselves. Cells both respond to their environment and create that environment. So you can't understand what's happening in a tissue by looking at cells by themselves. And so you have to think about single cell behaviors, cell-cell interactions, cell environment interactions, and the kinds of physical constraints that limit what's possible. In this case, Paul is thinking specifically about multicellular cancer as an ecosystem and what are called the hallmarks of cancer, but the same kinds of questions apply in development, in infectious diseases, and in tissue engineering and toxicology. Virtual tissues are a term that was developed originally by the EPA, and these are mechanistic and dynamic agent-based multicell models. That's what we're going to be doing. They're focusing on emergent spatiotemporal phenomena resulting from the interaction between those agents, typically cells. They're mechanism-driven. As you'll find, they're additive. You start with nothing, and you add things to your models. You don't get anything for free in these models. And they're based on quantitative submodels of what those typically cells and other components do, how they appear and disappear, what forces they apply, how they change shape, how they move. And with a simulation environment like CompuCell, we'll typically be modeling hundreds to hundreds of thousands of cells, which means it's about like having a 20x objective for a microscope. And typically we'll have movies as our outputs. If we think about tissues and living organisms as an engineering problem, and we want to be able to, say, design a drug that's going to do something, we face a problem. When we do engineering, we're taught that the measurement and the control that we apply should be done at the scale at which we want our outcome to happen. In biology, we have a problem. Most of the high-frequency measurements we can do are things like blood chemistry, which are averaged over the whole body. Most of our interventions, surgery is an exception, are drugs. Those act at a molecular scale. The outcomes we care about are in the middle at the tissue and organ level. And so we've separated measurement at large scales from control at small scales from the things that we care about at intermediate scales. And that means that control as a biological problem is a very difficult one. And so we use virtual tissues to integrate across scales to try to be able to predict how a change at the molecular level is going to propagate up and down, because propagation goes both ways, to give us the outcomes that we want or potentially don't want. And the analogy I always like to use here is that omics gives us parts lists, but if I gave you a list of all the parts in your car and asked you to tell me how good the car is to drive, you'd have a hard time doing that. And so these kinds of models essentially are taking those parts lists and trying to build a model of how the car drives. One of the things that we find very much in biology is that cells move, especially during development, but also in wound healing and in normal homeostasis. These are a variety of experimental movies from various sources showing developmental phenomena. Top right, we have the nuclei stained in every cell in a zebrafish embryo. The bottom left is the growth of blood vessels between the somites, the segments, in a zebrafish. The bottom right is the formation of those same somites, segmental structure in a chick. Upper left is branching morphogenesis in vitro. So we can't assume that cells don't move, and we can't assume that cells don't interact with each other. And so virtual tissues, again, are designed to explore these interactions between scales. Typically, we will have some kind of assumption of control inside of individual cells. Sometimes we'll assume that cells aren't changing during the time we're doing simulation, in which case we may not have that control. We'll have those control networks change what the cells do. Those cells will then do things in an environment, interacting with each other, secreting and absorbing chemicals, responding to extracellular forces. And this is a simulation of that segmentation in a chick. In one case, in the middle, we have extracellular chemical fields. On the right, we have the gene expression in individual cells, which oscillates. The left, we have the reorganization of cells and their differentiation. Often we will put these kinds of models inside a larger non-spatial pharmacokinetic model if we want to do things like drug dosing studies to know how much drug gets into given organs at a given time. These kinds of models have been used to understand basic biology, development, homeostasis, disease, to predict the effects of drugs and chemicals, either in good ways in drug design and discovery or bad ways in toxicity, to design and personalize therapies. And again, these are particularly useful when you have a lot of spatial heterogeneity, which is almost always in biology, or when you actually have cell movement or reorganization, or cell death or cell birth. On our CompuCell website, you can see recent papers. You can see that lately people have been using CompuCell to train neural networks a lot. People have studied cancer spheroids using CompuCell, migration of individual cells, neural tube closure defects, that's developmental toxicity, and you'll find many, many other kinds of applications. If we just have a bit of a zoo of CompuCell applications, here on the top left, we see a single cell modeled in quite a bit of detail, crawling. You'll see perhaps that simulation later in the week. On the right, we see a simulation of a vascular tumor where the tumor is growing. The red are the blood vessels. As the tumor becomes hypoxic, the cells in the middle don't have enough oxygen. It sends out a signal causing the blood vessels to proliferate, angiogenesis, and that proliferation then leads to further growth of the tumor. The bottom left were studies done to try to optimize therapies for diabetic retinopathy. so models of individual patient retinas. Bottom right were simulations of invasion of the retina by blood vessels during age-related macular degeneration, so-called choroidal neovascularization. This kind of modeling has been used a lot, as I mentioned, in toxicology. You'll see the same picture in a few minutes again. Studies from the EPA of vascular disruption, urogenital disruption, disruption of the palate, the roof of the mouth, from various kinds of chemicals. In these cases, they have quite detailed models of the chemical signaling inside of each cell and between cells, and mechanical models of what's happening at the tissue level. You'll hear some talks about that in the breakout sessions later. Our group did quite a bit of modeling of the liver and toxicity in the liver, modeling flow in the liver, trying to understand variability between individuals in terms of response to particular chemical toxicants. You can also model in detail the actual pattern of flow in the liver to get what's called microdosimetry or oxygenation in the liver. And there have been some beautiful talks about talks in the past. I don't know if we're having one this year on modeling the details of blood and flow and metabolism in the liver with these methods. A talk that you will hear is a model of the immune system. Starting in 2020, people used this kind of modeling a lot for modeling the immune system. This is work modeling the maturation of B cells in lymph nodes using CompuCell. Again, there's a signaling model outside the cells. There's a signaling model and a gene regulatory model inside the cells. The cells move in the lymph node. They differentiate and proliferate and die in response to their environment. Something that we've always liked was that it was actually the person who was the first author on this paper was a high school student when he did this work. And he came for many years and gave talks about it afterwards. So we were quite pleased that a high school student was able to do major publishable work using CompuCell. So we hope that it'll inspire people to do that. This is recent work in our group looking at injury in the cornea, surface of the eye, and recovery after injury. We may hear a little bit about that later in the class. As I mentioned, people have done a lot of modeling of infectious disease. This is a model of influenza in the lung, quite a detailed model with many, many cell types, many chemical signals, virus introduced at a low rate and then spreading through the tissue. And this was a model that was validated in quite a bit of detail against time series measured in animal models. This kind of modeling has also been used to try to understand drug dosimetry for antivirals. Beautiful work that was done in the past year or two from Megan Haas at University of Virginia was looking at muscle fibers, damage and recovery. She started her work in this area by taking this course a few years ago. And this was her PhD work. She was interested in how estrogen in women before and after menopause or after menopause with hormone replacement therapies affected the rates of muscle damage and recovery. Again, she built a very detailed model of all of the cell types in muscle and many, many different growth factors and immune cells that affect damage and recovery. The first of her papers came out last year. You can find it here if you're interested. So that gives you a little bit of a flavor for the different kinds of things that you can do with this kind of modeling. Certainly one dream is to use what are called medical digital twins, that is detailed predictive models, to personalize medicine and make it faster and more responsive. That may take a while, but if we are ever going to be able to use formal methods to diagnose, predict, and optimize treatment, then we have to be able to say how health states are going to evolve. And this kind of modeling is an important part of that. This kind of modeling is also very important in something that is, as people know, the world of research, especially in the United States, but also in Europe, is changing very rapidly. And central to that new approach is what are called new approach methodologies. Sometimes NAM is a universal acronym, but in fact, how it's expanded is differs with different people. But new approach or novel approach methodologies are methods that try to extrapolate from in vitro culture or organoid culture to humans directly without using whole animal models. And if you look at the NIH announcement from a little while ago, you'll find out NIH to prioritize human-based research technologies. And here, bullet point number 2, maybe too small for people to read, computational models which simulate complex biological human systems, disease pathways, and drug interactions. Now, it happens that the person leading this effort at NIH, Nicole Kleinstreuer, was the first author of a CompuCell paper some time ago, predicting chemical toxicities from drugs in angiogenesis. And so Nicole Kleinstreuer, Dr. Kleinstreuer now, is leading the effort at NIH to try to use this kind of modeling. And I don't mean CompuCell per se, but this kind of modeling generically. And so this is going to be more and more important if you're going to be writing NIH proposals in the future, or for that matter, NSF proposals. They're going to need to have a computational component. Virtual tissues are very helpful in many ways. They can function as variable power microscopes to allow you to change the resolution at which you look at problems. They allow you to control graded perturbations in ways that are hard to do in experiment. You can test mechanistic hypotheses and infer parameters. They reduce the need for animal experiments, which is what I was just talking about. They allow you to test situations that you wouldn't be able to realize in the real world. You can do many, many more combinatorics than you'd be able to do in a typical experiment because simulations are cheap. They're great ways of visualizing, communicating information and knowledge. And they're a great way of codifying and consolidating our understanding of problems. They're ways of reusing and integrating knowledge and ways of extracting more information from existing data. We can also do things like explore counterfactuals. There may be things that never occur in nature that we'd like to be able to study that we can do in experiment. Building models is hard, it takes work, and it codifies and helps us understand what we don't know. So very often you discover when you build models that there are experiments that need to be done that you wouldn't have thought of if you hadn't built the model in the first place. It's not always easy. Biology is copious and heterogeneous. You've got lots of components, lots of different types of components, lots of parameters. Those parameters may not always be obvious. And you can't describe things in complete detail. And as we'll find out, it's not always easy to compare experiment and simulation in a quantitative way because biology is stochastic and our outputs are movies. There's a lot we don't know. And we have tight coupling between components, and that makes the simulations in some ways more complicated than they might be otherwise. And so we have to be humble when we build these models. And one of the hardest parts of model building is to decide what to leave out when we build models. We can't include everything. When we started out, CompuCell3D was the only virtual tissue environment around, pretty much. These days, there are lots of great tools. I'd call out Morpheus, Physicell, and TissueForge, Artisto, and Chaste as tools that are of great interest. We work closely through OpenVT, which is a community trying to disseminate these model methods and trying to make them more compatible with each other. so that eventually you'll be able to take a simulation written for CompuCell and run it in one of the other packages. At the moment, we can't do that, although we can help you translate between them if you want. And if people are interested in this problem of exchange between modeling methodologies, we'd love to have a discussion with you. There are many different ways to represent cells. They're what are called center models, which are point-based. Those let you have lots and lots of cells in your simulations, 500,000 cells or even 1,000,000 cells, but they don't have any spatial detail or limited spatial detail. There are also models that have more explicit cell shapes. You can cluster point particles together, as in molecular dynamics. Those are called subcellular element models. You can make what are called vertex models. You can make very detailed finite element models, where you actually try to get all the mechanics right with solids modeling. Karniadakis at Brown and Kasiresniak at Moffitt are famous for doing those kinds of models. And there's a HAL package from Moffitt that implements one of those. We're going to be focusing on methods in CompuCell. CompuCell has been around now for almost 30 years, 25 years now. You're going to have an editor called Twidit and Player, which is a simulation environment. CompuCell lets you build these models at multiple scales, and it runs on a variety of platforms, we hope. I hope people are able to get it to run on your platform. Windows, Mac, Linux, and it's also cluster and cloud executable. And next week, you'll hear a little bit about that aspect of CompuCell. CompuCell tries to build in support for key biological and physical concepts. There are fields, mainly chemical fields, and they do things like diffuse and decay. There are cells and generalized cells, which represent cells and their regions and substructure. There are links, which are going to represent things like philopodia and junctional adhesion, and then there are going to be a variety of networks that can be used to control cells inside the cell to represent gene regulatory signaling and metabolic networks, or outside the cells to represent things like chemical transport through the body. We're going to have Friday and then again next week discussions of network modeling. Specifically, CompuCell supports SBML models, antimony models, MabBus, Boolean network models, also supports CellML, and it also supports the rule-based package BioNetGen. If people, we're not going to be teaching BioNetGen, but it's available for people if you like it. Now we're going to start by actually running a little simulation. And so I'd ask everybody to fire up CompuCell and follow along with these exercises. You're going to build this simulation later in the week. Today we're just going to run it to get familiar with using CompuCell Player. If you're using a Conda install, you can download the demos if they didn't come with the package that you did, using the link here in the chat. If you installed CompuCell with the regular one-button installer, you should have it, the demos already with you. And let me wait and make sure that everybody has CompuCell running. So CompuCell comes packaged with a number of demos. These are small simulations that illustrate biological principles or modeling or programming concepts. And we're going to run the 3D angiogenesis simulation. Again, CompuCell consists of a player for executing simulation, a Twidit, a model editor. You're also certainly possible to edit CompuCell if you prefer something like VS Code or Visual Studio. But today we're going to be using a Twidit. Twidit, we find, is a little bit easier for people who are beginning with CompuCell. If you're a very experienced user, then VS Code or Visual Studio has some features that are very nice as well. So in our very first exercise, we're going to run an angiogenesis, vasculogenesis simulation. This goes back almost 20 years to work that was done by Roland Merckx, who's now at Leiden University as the head of the Lorentz Institute there. And this was work trying to understand the mechanisms of vascular patterning, particularly capillary development. And he was interested in trying to understand how capillary cells, vascular endothelial cells, organize both from sprouting angiogenesis, when you already have blood vessels, and in vasculogenesis, when you have dispersed cells to begin with. And these simulations are going to be implemented in CompuCell3D ML and Python. I should say that the code that he wrote back in 2005 wasn't CompuCell, and actually it turned into a package that they develop now and distribute out of Leiden called Tissue Simulation Toolkit, which I definitely recommend you taking a look at. So we're going to start out with endothelial cells, and we're going to ask how they organize. And there are some experimental observations about this. And those experimental observations suggested, one, that the cells secrete a growth factor, a morphogen called vascular endothelial growth factor A, VEGF-A, and that the cells both secrete and respond to it. And they respond to it by moving up gradients of vascular endothelial growth factor A, VEGF-A. That's called chemotaxis. And so this is an autocrine or paracrine chemotaxis. There were also experiments with our collaborators at Kansas University Medical Center that showed that chemotaxis was inhibited when vascular endothelial cells touched each other. That's through a molecule called VE-cadherin, vascular endothelial cadherin. Cadherins are cell adhesion molecules. And that if you interfered with VE-cadherin, you didn't get vascular patterns, you got blobs. And based on that, we developed a model of autocrine chemotaxis. Here with contact inhibition, and you get vascular patterns. You turn off the contact inhibition, now the cells respond on all the surface and you get blobs. Up on the right, you see actually an experiment using human umbilical vein endothelial cells forming this kind of vascular pattern. The simulations showed that the chemotaxis has to be very short range. The diffusion length of the cabicle has to be very short range. And so the key physics is that the cells move randomly. There is a chemical VEGF that diffuses and decays in the environment, and the cells move up gradients and secrete that chemical. And so now we're going to run that simulation. So reminding you, on Windows, if you installed with the .exe installer, search CompuCell, your search tool, and you should be able to launch Player. On a Mac, after terminal installation, CompuCell is in the user, whatever your username is, CompuCell3D folder path. Note that it is not in the application folder by default. You can search for CompuCell using the Finder Apple F command. To launch CompuCell, double-click on the CompuCell3D command file, and you should see a terminal window, and then Player should open. If you installed via Conda, you're going to have to activate your Conda environment. Conda activate, whatever the environment name you gave it was. Once you're inside of that, Python-m cc3d player 5. The very first time you launch CompuCell, it may take a while to launch. Don't close the terminal window that appears during the launch. If there are any warnings, sometimes both Macs and PCs will say it's an untrusted piece of software. You may have to give it permission to run. I'd say today that in this morning one, if you can't do all of the exercises or something doesn't work, don't worry too much. This is really a run through everything we're going to talk about right now. We're going to go over again more slowly and in more detail. This is really to get an initial familiarization with the capabilities of CompuCell. Again, you'll get, if you miss something now, you'll have the opportunity to come back. Okay, it looks like the vast majority of the people here have CompuCell running at this point. Okay, so I'm going to now give you a quick introduction to CC3D Player. Again, you'll have plenty of time to consolidate this, so don't worry if you don't get through everything that we do right now. The first thing we're going to do is open a demo and run it in Player. And this is a bit of an experiment because CompuCell has a new feature in Player, which is called Open Demo. If you installed with Conda, this may not work because the path to the demos may not be what you expect. If you installed with Conda and you don't have demos installed by default, please go to the SourceForge CC3D files 4.7.0 demos.zip and download it and unpack it. And the instructions are given there. In Player, you will find a button called File at the top of your screen. Pull down that menu, and you will see a line that says Open Demo. If this doesn't work, don't panic. I will give you an alternative way of doing this in just a second. But for the people for whom it works, it makes life a lot easier. So we're going to try this first. You will open up a demo browser, and you'll select Angiogenesis. That's the simulation we want to run. Again, if it's not working and you used a one-button install, then you might be in an earlier version of CompuCell. Again, I'll give you an alternative way of doing this, but this is easier if it works for people. If you're using a Conda install or you're on Linux, you have to download the demo separately, and so you have to follow the instructions that I gave you earlier. Once you've selected that Angiogenesis demo, you should see Demo Browser. It should pop up something that says Description, Python, XML. If you select the description tag, it should give you a little information about the model. If you select Python, it will show you the header of the Python code. XML, it'll give you the header of the XML code. Once you've selected Angiogenesis, you click Open this demo. Again, if it's not working, we're going to have another way of doing it. Somebody says they don't have the Open Demo button. That's either in an earlier version of CompuCell or you're not in Player. Okay, so it looks like about half of the people got it to work this way. If it didn't work that way, let's do it the old way. The old way to do it is you can either click on this up arrow button here in Player, or you can pull down the File menu and select Open Simulation File. Nothing should happen when you click Open the demo. The dialog box will disappear, and you'll see Player sitting there. I'll show you what that looks like in just a second. And I can maybe this is a time actually to do a split screen. Let me finish going through this with you, and then I'll share screen and I'll do it on the screen for you, okay? Once you've gotten to Open Simulation File, or you've touched the Open project here, you'll get a dialog box. You will have to navigate to wherever you installed your demos. Classically, on a Windows machine, that would be CompuCell3D demos, and it's a little bit complicated. Book chapter demos, computational methods in cell biology, angiogenesis. Select that. You select the .cc3d file and hit Open. When you're going to edit a simulation, which we're not going to do right a moment, you're going to have to make sure that you've put the simulation somewhere where you have write access. It's possible that you've installed CompuCell in a place where you don't have write access. In that case, when you try to run from that directory, it'll crash. Once you have it loaded, you will see in Player 3 buttons at the top of the application. There is a play button, like on an old CD player or cassette recorder, that will play the simulation, run the simulation. There is a circle, which is a step button, which will do one time step in the simulation. There's a pause button, which will allow you to pause and restart the simulation, and a stop button that terminates execution. In general, you don't want to hit stop unless you're certain you don't want to restart, because when you hit stop, everything goes back to the beginning. I'm going to just do this on my computer. I'm going to launch CompuCell. You should see the Python command prompts. And here Player is launched. I'm now going to go to File, Open Demo, select Angiogenesis. It says this file doesn't have that kind of file available to preview. If I click on description, I'll see description of the simulation. This is an all Python simulation. And so if I click on Python, I can see the code there. I now click open this demo. And as somebody pointed out, that doesn't actually do anything. If I want to run it, I can hit step, and that will start the simulation. You should see this. I'll explain people how to do 2D versus 3D visualization in a minute. If I hit run, the simulation will run. Pause, step. Okay. People who've already got it working can try playing with the visualization and using the step and the run and the pause buttons. If I wanted to open the simulation manual, I would go to View, Open Simulation File. Here I have to stop the simulation, Open Simulation File, and then I would have to look for demos. And I can look for book chapter demos, Angiogenesis, CC3D. I'll show you how to do the visualization in a minute. Don't worry about the layouts and things. I'm going to go over that in just a second. So when you are using Player, you have to learn how to manipulate Player. And there are a number of things that you might want to do. You can resize the Player window as a whole by pulling on the corners. You can resize the component that's called Model Editor here on the left versus the display window on the right. Within Player window, you may have multiple sub-windows. This will look a little bit different on a Mac because the windows will not be captive on a Mac. They'll be free-floating. And you can use minimize, maximize, and close. I strongly recommend not closing sub-windows in Player because it can leave it in an illegal state. So you can try using those a little bit. Now, in the unlikely event that the simulation crashes, and it's an existing simulation, which we know is has the correct syntax, that can be because Player has been left in an illegal state. And this is more for future reference. You shouldn't be having this problem now, but there's something called an SQLite file that specifies the Player configuration. Unfortunately, that's a hidden file. And so you may need to search for _settings.sqlite in your simulation folder. And if you delete it, it will reset Player configuration. If Player still crashes, there may be a problem with the master SQLite file. In that case, go to users, your username.CompuCell3D_py, and delete the .sqlite file there. You shouldn't have to do this, but in putting it in the record so that if you have this particular problem, you know where to find that information. There are a couple of other things, especially if you have a multi-monitor configuration. If you open player on an external monitor and then disconnect the external monitor, player or TwidIt may try to open where the external monitor was. In that case, reopen player and TwidIt with the external monitor present, move them to your main monitor, close them using the stop function, and then disconnect the monitor. Sometimes the pull-down menus will appear in odd places. You may have to look for them. That happens to me, especially when I'm using Zoom. They're there, but you may have to look for them. On a Mac with 4.7, sometimes the pull-down menus will show up, but they'll seem to be empty, and that is a configuration issue. You'll have to go to Settings and select the default theme. The light and dark themes may not work on some Macs. So now let's talk about controlling cell visualization. In that simulation that we just loaded, hit the step function, and you should get a display. Depending on exactly which simulation you're running, you may get a 2D display like this or a 3D display like this. You can switch in the graphics window between 2D and 3D. You click here where it says XY, and you can select XY, YZ, XZ, or 3D. Notice that you will have to pause and restart your simulation to change the display in that way. FPP plugins is a warning. You don't have to pay attention to that. It doesn't do anything. The drop-down is there. It's hiding somewhere on your screen, so you may have to look around for it. If you are on a Mac, you will have to go to Settings here, Configuration, Setup, and then under Theme, select Default Theme. Okay, then you should be able to see your pull-downs. I notice here, if I do that pull-down shows up on my second monitor, not on the main screen. So you're not seeing it. When I do this pull-down, it shows up on my other screen. So was the question, how do I fix? Okay, to fix the settings here, the settings is under configuration here, or I can do tools, configuration like that. I select the first tab, setup. I go to theme, and I pull down and select default theme on Mac and hit OK. I'm going to use dark theme. Actually, I'm going to use light theme for the moment for myself. I saw a bug, Hayden, in the default theme for Windows. She'll have to report. Yes, the colors may well be different for you. Don't worry about the colors. The graphs may also be different. And the scales may be different. One issue is that Player remembers what you've done with it. And since I've been using Player, the settings will be slightly different on my computer from yours. Somebody says you can't get 3D if you pull down this menu. Again, you have to look for it. You have to select 3D. As I say, on my computer, that pull-down menu shows up not on the screen that I'm sharing, but my other screen. If you hit play, you should see this. Now, there's a nice feature, which I was going to come to in a minute, but we can talk about now. It's only available on Windows machines. Macs don't allow this, unfortunately, which is if you go to Window and you hit Tile, it will lay out the screen again. Now I'm going to move on because we've got a lot still to cover. You'll have lots of time to play with these things on your own. Again, here's this comment about Mac issues. This will be in the notes. Settings, setup, default theme on Macs. You're able to open as many graphics windows as you want. So watch the simulation run. You should be able to rotate, zoom, pan. If you get things messed up on your screen, hover over it and hit R, and that should reset the screen. So you restores it to its default settings. If you're on a Windows machine, pull down Windows, Tile, and it will lay out the screen for you. Unfortunately, that's not a feature on the Mac. In the Mac, the windows aren't captive. You have to lay them out by hand. Okay, let's talk about cell visualization. There is a menu called Visualization, and you have some tick boxes, cells, cell borders, cluster borders, which we're not using at the moment, concentration limits, CC3D output. Typically, we're going to represent cells. In 3D, we often don't want to represent cell borders. When you make major changes, the simulation will pause. You'll have to remember to hit play or step after you've made the change to resume. If we go to configuration and we select cell colors, you'll get a window that allows you to select the colors of the different cell types. You always have to remember to hit OK to save them, to accept those changes once you've made them. And you can try that. Why don't you try changing the colors of your cells? To change the color of the cell here, select configuration, cells/colors, select the cell type you want to change. You can select the cell color, and you can also select the cell border color if you like. Try changing that a little bit. I'll give people just a minute to try those changes. Okay. As always, we'll say this many times, if you get ahead, feel free to explore. Don't feel that you have to wait for the instruction to catch up with you. There are lots of things to try. We can't cover absolutely everything in class. And so there are all sorts of wonderful things to explore in CompuCell. One of your windows should be chemical field. Visualizing chemical fields in 2D is pretty straightforward. In 3D, it's a little more subtle. I'll show you in a second. If you want to do 3D visualization of chemical fields, you go to Configuration, you select Field, and then within Field, you select which chemical field you're using here. You're going to typically set the minimum to 0, maximum float, and you'll select contours and how many contours you want. And this is the same chemical field seen in 3 ways. 2D without isosurfaces, without contours, 2D with contours, 3D without contours, and 3D with contours. And let me actually do that for you. If you want to add chemical fields, this was the question that was just being asked. I originally had this slide a little bit earlier, and then I thought I would show you how to use things before I added windows. If you click on this 2-window icon, or pull down window, new graphics window, it'll give you a new window. Then you have to say whether you want to display the cell field or the chemical field. Let me do that with you. Okay, I'm using that open angiogenesis simulation. I'm going to hit play. And I've got my windows again. Again, R for resize, reset, 3D, 2D, 3D. And now I'm going to go under Visualization, Configuration, Field. Here I have isosurfaces turned on. I'm going to turn isosurfaces off, hit OK. This is what I get in 3D, not so pretty. 2D, clearer. turn isosurfaces back on. Visualization, configuration, field, show isosurfaces. Okay. Go back to 3D. Okay. You're going to have a lot of time to play with CompuCell later. So this is just a first attempt to do. The next thing that I would like to do is to show you TwidIt, the model editor. And we're going to use TwidIt and Wizard to create a cell growth simulation. This is a very simple simulation, but we'll learn a lot about CompuCell doing it. So to launch TwidIt, we can use Finder, or if you have Player open, you can launch TwidIt by hitting this little pen and paper icon here, or under File, click Start TwidIt. So I want everybody to launch TwidIt and give me a thumbs up when you have TwidIt running. Inside of TwidIt, we are going to start a new simulation. You will go to CC3D Project, pull that down, select New CC3D Project, and you'll get a box that says Simulation Wizard. Now you'll type the name of the simulation, and please be reasonable about file names. Don't begin the file name with a number. Try to avoid special characters. Try to avoid spaces. You can call this Cell Growth Simulation 1 or something like that. Typically, you want to put this in a directory which you have control over. A good choice would be your desktop for the moment. Once you've selected the folder you're going to put it in, hit Next. I can do this with you after we've walked through the steps. Everybody got that? Thumbs up from people if they got that working. You don't have to close Player, but you do need to be able to see TwidIt. So depending on how your screen is laid out, you want to make sure TwidIt is visible. You definitely can have them both run at the same time. We normally do. Okay. Now, the next window that you're going to get, and this will be slightly different in 4.6 and 4.7, is going to say General Simulation Properties. And you can just hit Next. You don't have to change anything. Something that's new in 4.7 is you can specify the time units. 1 Monte Carlo step equals 1. And you can say 1 minute here. And you can say 1 voxel or lattice site equals 1. And you can say microns here. It's not necessary to change anything. So again, you don't have to change anything in this window at all. Just hit Next. If you want to make the units what they are, that's fine. That units option won't show up in 4.6. The next window is specifying cell types. We're going to need 2 cell types. Type cell 1 and hit Add or Return. And then type cell 2 and hit Add and Return. Again, don't be perverse in your cell type names. Don't begin a cell type name with a number. Don't use special characters. Don't use spaces in the name. Then hit Next. If you are in 4.6, the next window you'll get will be chemical fields, and you'll hit Next. In 4.7, the next window you'll get is cell properties and behavior. So in 4.6, hit Next, and you'll see cell properties and behavior. In 4.7, this is the next window you'll see. Tick box contact, growth, mitosis, and volume. Make sure all 4 of those are checked. Everybody got that? It's easy to forget contact and volume, so it's important that you collect those. And I have a thumbs up on that one. Okay, and hit next. And I'll do this with you in just a second. You'll almost always use those options, but those options will vary depending on the simulation you're running. In 4.7, after I hit next on cell properties, I'll get the chemical fields. We're not using chemical fields, so you just hit next. You'll get a window that says configuration complete. You hit finish. You should see TwitIt. It should be blank for you with, in the top left, modules, whatever you called your simulation here. Double click on that, and it'll open 3 tabs which contain the simulation. If you selected, there's a question, if you have selected growth and mitosis stepables, the only choice you will have will be volume local flex. So I'll walk you through that now in a second. When you save, you can use the down button to save the simulation as a whole, or the double floppy disk icon to save the simulation as a whole. If you want to rename things, you select Save CC3D Project As. I strongly recommend not using the single floppy disk save options because they save parts of the simulation, not the whole thing. I always use the full save all. There are some special cases where you want to save part of a simulation and not others, but we're not going to have that run into that very often here. Okay. Once we have the simulation defined, if player is running already, the little CompuCell icon will be grayed out. If it's not, you can hit that. You can pull down CC3D project, select open in player. You can select in the left-hand navigation pane the top level name of your simulation, right click and select open in player. When you go back to player, if you have another simulation running, you'll get a warning, simulation still running, current simulation still running. Do you want to stop it? You set yes, and it'll overwrite whatever you were running with the new simulation. Okay. And as I mentioned before, if we've saved the simulation we created, we could go navigate to that, open with the up arrow, or file, open simulation, and run it from wherever we saved it. All right, let me do that for you in front of you as well. I'm going to go here. I'm going to switch to TwitIt. And since I have TwitIt running, I have to find it. Where did it go? Where did my TwitIt go? Okay, it's taking a second to load. Okay. So clicking that pen and paper icon, launch TwitIt. Here's TwitIt. I don't have anything running in it. I'm going to go CC3D project, new CC3D project. I'm going to call this cell growth. And since I've done this many times, test, I'll call it version 4. I happen to be pointing to my desktop already, so I'm going to leave it there. Next. I'm not going to change any of the defaults for simulation properties, although as I mentioned, I could say that this is 1 minute, 1 Monte Carlo step is a minute. I can say that. 1 voxel is a micron. I can say that. Hit next. Yes, depending on the theme that you've selected for your player and your TwitIt, you may get different color options. You may get dark or light. Okay, cell types. Cell 1. Cell 2. Next. I'm in 4.7, so I get the cell properties. Next, I select contact. growth, mitosis, volume local flex. Next. I'm not going to use chemical field, so I hit next. Configuration complete, finish. People see that? Now I double click here on cell growth and I see the simulation. If I want to run this simulation, I can click here, open in player. I'm now going to switch back to here. It should be giving me a message saying to stop it. See what happens if I restart. No, it hasn't picked it up, so I'm going to close player myself. I'm going to go back here. Oh, it's created a new window. All right, created its own. Here, I've got my new simulation. I'm going to resize my simulation here. You'll notice here at the top, it gives me the name of the simulation it's running, cell growth test. I realize the font's pretty small. On this screen, I have a high resolution screen. If Twidit is crashing when you hit save, it probably means that you're pointing to a directory you don't have write access to. So when you create the simulation, you have to be sure that you select a directory you can create the simulation. Okay, how many people are able to get this to run? Can I have a thumbs up for that? Again, we're going to have plenty of opportunities to debug things with people one-on-one. Okay. All right, good. Now, I want to do a little bit more reminder about how you manipulate CC3D player, moving the boundaries. We've seen that before. Remind, stop, step, pause, stop, start, step, pause, stop. And that's a simulation. Now, we haven't talked about what that simulation does, and I'd like to spend the rest of our time talking about what that simulation does and have you run one or two tiny exercises to play with that. Okay, so this is going to be discussing CompuCell specifications. Again, we're going to be spending all week on this. We're going to change the cell growth rate and the initial condition in this specification. And we're going to go a little bit over what CompuCell looks like. CompuCell simulations generally have two parts, a CompuCell markup language part that specifies static aspects of the simulation and a Python part that specifies dynamic aspects. Some simulations are markup language only, some are Python only, most have both. Let's go back and look in Twidit at the simulation. When I double-clicked on module, well, here it's called Module 1 Simulation, but I called it Cell Growth Test 3 in my case. You'll see three tabs. One tab will be XML, one tab will be .py, and one tab will be steppables.py. That middle tab, we almost never use. What's called the main Python script, we almost never use. That's created automatically in most cases. We will be editing the XML and the steppable. We have the scroll bars to go through things. You could change the size of the text and various other things like that. Let's look at the XML. The XML comes in blocks. And probably screenshotting the dark format wasn't the best thing for this, but that's what we've got there. And I'm going to walk you through a couple of different things in the XML. There will be line numbers by default. You can turn those off. The line numbers may be slightly different in your code. The first thing that we have is code that specifies the size of the simulation. You'll see CompuCell3D revision 0, in my case 4.7.0. You'll see a tag called metadata. number of processors because CompuCell is intrinsically parallel. If you have a lot of CPUs on your cores on your CPU, you can run more than one processor at once. Debug output frequency, which you typically don't play with. In the new versions of CompuCell, you have the conversion factors of Monte Carlo steps to minutes and length to microns. Then we have a block called POTS, which says the dimension of our simulation, in this case 256 by 256 by 1. It's a 2D simulation. Steps, and some other things we're not going to worry about right now. The next block is the cell types. And here, remember we typed cell 1 and cell 2. Cell type ID equals 1, cell 1, and so on. These are called plugins. Plugins specify cells and chemical field behaviors. And those are actually things that are implemented in C++, compiled C++. Unless we specify cell types, we don't have them. By default, there's a special cell type called medium, which has some unusual properties, sometimes helpful, sometimes a little bit annoying. We'll come back to that later. Remember, we said the cell has a volume, and there's something here called plug-in name equals volume, saying that that's true. Again, here we have cell type ID 0, medium, cell type ID 1, cell 1, cell type ID 2, cell 2. If your player is working, you'll have in the lower left of player cell 1, cell 2, and the colors associated with them. Might be nice, if somebody really wants to be energetic, to be able to add the color information to the cell type specification, the XML, something we've talked about for a long time. The next block of code is called contact, and that specifies how cells stick to each other. And in this case, we have a plug-in contact, and it says medium sticks to cell 1 with energy 10, medium sticks to cell 2 with energy 10, cell 1 sticks to cell 1 with energy 10, and so on. We'll ignore the neighbor order command for the moment. There's a little bit of a trick here. Bigger numbers mean less cohesion, and medium has special properties. So actually a medium cell energy of 10 actually corresponds to a cell energy of 5. This means that, I said that exactly backwards. Medium cell energy of 10 means that it corresponds to a cell energy of 20, I'm sorry. And that means that cells stick to each other more than medium, so that the cells are going to stay compact. You can actually change those values in player as the simulation runs, and I'll show you that in a minute. The next block that's important to us is called a steppable. Steppables are only executed once per time step in the simulation. Plugins are executed more frequently. And here we're doing a very simple initializer, uniform initializer. It draws a rectangle of cells. Box min is the lower left-hand corner. Box max is the upper right-hand corner. Gap specifies, and it draws squares of width 7. Gap says that there's no space between the cells. And types would specify what kinds of cells that we want. When you edit, twit it, and hit Save All, player should, when you hit Stop and Start, automatically update the code. If it doesn't, close player and reopen it. Try going into your code, finding Uniform Initializer, and change the value of box min to be just a little bit smaller than box max to see if you can create one cell to start with. Width is the size of the squares that it's going to draw for your initial cells. Okay. Is that little exercise clear? So let me actually do this a little bit with you. Let me find Twit-it. Here's Twit-it, my simulation. Here's player. I'm going to restart the simulation. This is the initial configuration that I get. I get lots of cells. But if I want this to be a reasonable tumor simulation, I actually probably want to start with one cell. And so what I'd like you to do is go to Twit, find the XML, find uniform initializer, and go and change box min values so you only draw one cell. You can leave box max alone for the moment. Make things a little bigger so they're easier to see. You can zoom in and out with view here. Okay. Yeah, don't change the Z values. It's a 2D simulation. Anybody get that to work? I'm going to try it. I'm going to say, let's make this 200. 200. Hit Save All. I'm going to go back to Player. I'm going to hit Stop, Step, and I've got one cell. Somebody says player closes after stopping the simulation. That's possible. In that case, when you go back to Twit, looks like that may have happened to me too. Wonder what happened. I'm going to try relaunching. 4.7 is pretty new, so it's possible that there's some bugs in it. So if you find bugs in 4.7, please report them. People get something that looks like that. Yeah. Somebody says they had a cell that vanished. If your cell vanished, check to see that this volume plugin is there. If people forget to click that box when they created the simulation and they didn't load the volume plugin, it will disappear. Because if you don't say the cells have a volume, they don't. How many people were able to get that simulation run? Okay. Who needs help? So that looks like it's working for most people. Okay. Now we're going to talk about CC3D Python. Python specifies the dynamic part of the simulation. To do that, you click on this, whatever you called your file, it'll say _steppables.py. Click on that, or you can double click here in the left-hand margin. You'll see this. You'll see a variety of code here. You'll see some boilerplate from CC3D Core import, and you'll see class. If you don't know what a class is in Python, don't worry too much. And let's walk through the code together. Again, all of this will be discussed in great detail soon. Lorenzo says that in 4.7, player will occasionally crash on restart. Restart player, and it should be okay. Okay, let's look inside of the Python. You're going to be spending a week learning how to do this. The first thing you'll see is def start. At the beginning of every simulation, there is a start function that's run once, And we usually use this to initialize our simulation. You'll see 4-cell in self.cell list. That's one of the things that we do the most in CompuCell. 4-cell in self.cell list is a loop that applies to every cell in the simulation. If Python quit unexpectedly, it's possible that you introduced a typo if you edited the code, but you'll have to restart Python, and you'll have to restart player if Python quit. The next line is cell.target volume equals 25, cell.lambda volume equals 2. You're going to hear a lot about this afternoon, but cells don't have specific volumes or compressibilities unless you define them. And so for each cell, we're going to assign it an initial volume of 25 voxels, which would be 25. If I said to find the voxel size as 2, that would be voxel edge is 2 microns, so 4 microns squared, so 25 is a 100 square micron cell. Lambda volume is the inverse compressibility. The bigger lambda is, the less compressible the cell is. 2 is a little bit squishy, but it's not a terrible default. The next thing we'll see is the step function. Def step self comma MCS. Again, here we go through all cells in our list. Cell.target volume plus equals 1. Every cell grows at a constant rate of 1 unit per Monte Carlo step. That's really fast. So I would recommend changing that 1 to 0.1. So change the 1 to 0.1 in the Python growth steppable and see what happens. Hit save and then run, see what happens. And I'll do that with you. So here, I'm going to go into my Python. I have my start function, which I just showed you. I have my step function. And instead of 1, I'm going to make it 0.1. I'm going to hit save all. And now I'm going to go back to player. I'm going to hit stop, hit step. I have my new simulation. I'm going to let it run. In this case, like somebody else, the cell disappeared. So I'm going to try something a little bit different. I'm going to make that lambda volume here equal to 5. Make the cell a little bit more rigid. Let's come back and try it again. Looks like my Python crashed when I did. All right, again. There we go. All right, so there were 2 changes to make. As I mentioned, lambda volume equals 2 is a little bit too squishy for the simulation, so we'll make it 5. Target volume plus equals 0.1. Save. And we go back to the simulation. How's that doing for people? Okay, somebody asked, how do we make it centered? Well, let's think about that. Why don't you try that? Go back, look at your XML. Where did you put your initial cell? How would you change your value to make the cell be in the middle? Try that out as an exercise. Why did the cell vanish at low lambda? The answer to that is that we have a surface energy that is pushing on the cell. It's compressing the cell. And if you compress the cell too much, you can squeeze it out of existence. We're going to play with that later today. Okay. How many people were able to get the simulation to run at all? I see a couple of check marks. How many people didn't get it to work at all? As I say, we need something other than a thumbs up for the people who have an issue. Okay. There are a couple more things that the question of how do you start with one cell rather than multiple cells? Again, when you define the size of your box initially, if the cells are initially 7 by 7, depending on how big that box of cells is, you're going to have a different number of cells to begin with. The simulation started from the corner because of my box min and box max values. If I want to start in the middle, I would have to set box min and box max to be close to the center. It's 256 by 256. So if I start with something like 122, and 126, I should be near the middle. But again, you'll have plenty of time to try these out on your own. This is not meant to be an exhaustive. You'll have plenty of versions. In 4.6, the MCS to minutes doesn't work. Everything is purely in Monte Carlo steps. The fundamental units in CompuCell are lattice sites and Monte Carlo steps. The minutes and microns are decoration. I do want to walk through a little bit more of this code for you before we end. You'll notice that the cells divide. And cell division is actually something that's pretty important. And it's also pretty subtle. We're going to do that a lot. When a cell divides, you're creating a new cell, and you're going to have two cells where you previously had one cell. And in Python, you do that because in a little bit of a maybe non-obvious way, we first have to have a rule that says, when do our cells divide? And in this case, we're going to have the cells divide when their volume gets to be bigger than 50. Remember, their initial volume was 25. So the doubling volume is set to be 50. In reality, we might have a more complex rule, like looking at the cell cycle state. But here we're just going to say, if the cell gets big enough, it divides. Why don't we just divide the cell when it gets to be a certain size? In Python, there is a taboo. You cannot change the list you are iterating over. When we create a new cell by division, we change self.cellList. And that means we can't iterate over self.cellList at the same time that we're creating cells. And so what we do is create an empty list. Cells to divide equals empty list. We run through all the cells. We see if the cells are bigger than 50. If they are, we add them to the list of cells to divide. And then we go back, run through the list of cells to divide, and divide by. Okay. When we do division, we have four options. We can divide along a random plane. We can divide along a user-based plane. We can divide along the long axis or the short axis of the cell. Both of those happen in biology. We're going to leave this in random. When we create a new cell, we have to say what happens. Typically, we have symmetrical division, and so the volume of the cell is assigned half to the parent and half to the child. There's a function called update attributes that tells us what happens after a cell divides. Self.parent_cell.target volume divide equals 2 reduces the parent to half of the initial volume. Very powerful function, self.clone parent to child, gives the daughter cell all of the properties of the parent cell. And then, just for decoration, we say if the parent cell was of type 1, we make the daughter cell of type 2. If the parent cell was of type 2, we make the daughter cell of type 1. Remember, in Python, double equals is a comparison, single equals is an assignment. Slash equals divides the value by 2. Okay. So let's look at the code itself for a second. See this in the code. Mitosis steppable takes care of mitosis. Cells to divide equals empty list. If cell volume greater than 50, add to cells to divide. For cell and cells to divide, divide random orientation. Why don't you try changing the volume at which cells divide? You could make this 20 or 100. And you could also switch random division to one of the other options. When you do that, be careful about indentation in CompuCell. There's a nice block comment function under Edit, block comment uncomment, that helps with indentation. There are also these two indentation bars here at the top. I've done major axis here. And I'll make the target volume for division 100 instead of 50. Go back to my simulation, run it. See if it'll run. No, it looks just like Python. No, it looks like player crashed on me again. They'll have to restart. Oh, while it's relaunching, I'll make one other comment. In this case today, we either used demos or we used Wizard to create our simulation. There are going to be times when you're going to be downloading simulations. Simulations are not single files, they are directories, they're folders. And so typically we are going to distribute those as zipped folders, so everything is together appropriately. On the Windows machine, when you download a zip folder, you can put it anywhere you want, that's fine. On a Mac, the Safari browser usually unzips folders by default. And so it's important if it gets unzipped that when you move it, you move the whole file structure, not just the .cc3d file. I'd suggest rezipping them and then moving them and unzipping them where you're going to use them. It's critical that you keep all the files together and also the directory structure together. Both Player and Twidit can open zipped files, and they can also zip files for you. When you do that, you may get a warning like could not uniquely identify a project, and then you hit OK. The best thing to do to avoid that is to navigate to the place that you want to put that unzipped file. Somebody says in my XML plugin, contact started out with type 1 medium, type 2 medium, and no cells. You didn't create your cell types when you used Wizard. That's why that is. Okay, let's see if I can go back here. I'm going to try launching again. Play. Okay. So this is my version of the simulation now with the new larger doubling volume and the oriented cell division. You'll notice that the cells are now dividing along their perpendicular to their long axis. That was one of the division options. So people can try that and compare that to what happens when you have random division or minor axis division. You can make cell division asymmetrical by assigning different target volumes to the daughter and the parent cell. You'll see how to do that later in the class. Okay. Okay. So I'd like people to take a minute or two to fill out the Zoom survey. I'll ask Hayden to start the survey, please. Thank our funding sources again. Thank you for your patience. I really do appreciate people taking the class, getting started with it. And I know that the first lecture always is a little bit of teething. And I hope that as we go through the day, we'll go pretty slowly so that people have time to catch up. And everything that we've covered today in this lecture we'll go over in much more detail in the subsequent lectures. Please do fill out the polls. It's very important for us. And also, while we keep the polls anonymous in terms of your detailed responses, we do log people's poll participation. And if you want participation certificates at the end of the class, part of that is determined by how many polls you fill out. So we do need you to fill out the polls. So I'll keep this up for a minute or two. We can answer any questions that we have at this point. Thank you to the people who are TAing for this session. And we'll take a lunch break now until 1 o'clock. Okay, last call on the poll. Please remember to fill out the poll. We'll close the poll in 1 minute.